Hi, everybody. Thanks for coming to this Dean's Lecture. And as we know, it's Marsha Wills-Karp, who's chair of the Department of Environmental Health Sciences and professor in our school. And I think you also know that these lectures are designed for people who are either appointed or promoted to the position of professor in our school. And as I say, you know, this is a decision that's not made by me or actually by the A&P committee or the advisory committee. It's made by people outside the school, by people who write letters and people who are peers of our faculty. And so it's a high bar to get onto our faculty, to be appointed to our faculty, but it's an incredibly high bar to be promoted to professor. And I always thought of myself as a cynical, jaded person, and I was cynical and jaded when I was promoted to associate professor. But professor, it does make you take a pause and think about what an accomplishment it is. So you all know, I think, that Marsha came to us from the University of Cincinnati in the Department of Pediatrics, where she was a division director. But I'm just going to go through some of her formal accomplishments so that you know, to review her CV a little bit. So she graduated from Southwest Texas State University and then earned her doctorate in physiology from the University of California, Santa Barbara, and then did a postdoc at Yale. She came here as a postdoc as well from 1987 to 1990 and was on our faculty from 1990 to 2000. And then spent 12 years as director of the Division of Immunobiology, and she was the founding director at the Cincinnati Children's Hospital Medical Center. She also founded and directed the Immunobiology graduate program. And then in 2012, we were very fortunate to recruit her back here to the school as the chair of EHS. So as we're going to hear about, I think, and as you probably know, her research activities have focused on defining the environmental and genetic determinants of allergic airway disease. And she and her lab members have specifically explored the role of CD4 and Th2 and cytokines. And then, although it's not here, as I recall, she did seminal work on IL-13 and the importance of IL-13 in the pathogenesis of asthma. Since she's come to the school, she has also looked at the issue of the microbiome and gut, and I think there was an interesting article in our magazine about how transporting mice from Maryland to Cincinnati changed their phenotype and back again, and perhaps she's going to talk about that. But she's done work in bridging the schools here in the East Baltimore campus by creating an immunology lecture series, and more recently on the microbiome. Marsha's received many honors, and I'll just say a few. She gave the Rebecca Buckley Lectureship and the John Salvaggio Memorial Lectureship. She's been a member of the Faculty of 1,000 and served on numerous NIH study sections and is currently the chair of the NIH Allergy and Transplantation Review Committee. And recently resubmitted the EHS training grant in toxicology and got a score of 14, which is worth many honors, right? So that was something that we were all happy to see. And Marsha's authored hundreds of publications, and I think you'll agree with me that she's an insightful, creative researcher, and we're happy that although it took us a year between your schedule and my schedule, I guess more than a year, to schedule this Dean's Lecture, it's wonderful to have you back at our school. So please join me in welcoming Marsha Wills-Karp.
Thank you, Dean Clegg, for that nice introduction. It's a pleasure to be here, and as Mike told you, we rescheduled this many times. So unfortunately, in the interim, I've given similar talks in many of the departments around the school. So hopefully I'll have some new things to say to those of you who might have been at those seminars. But it's my pleasure to be back at the school and to work with all of you in many different capacities. Giving this talk is somewhat difficult as a basic scientist, because I know that many people in the school are not basic scientists, but are more population studies. So what I'm trying to do is to show you some of our basic science, but to really put it in context, and so that it'll be meaningful in terms of its public health implications. So hopefully you can let me know if I actually achieve that at the end of the talk. But since the late 80s, I've been interested in asthma, and as the title would indicate, over about the last 40 years, there's been what we would call the asthma epidemic. There's been tremendous increases in the number of cases, morbidity, hospitalization, medical care use, and deaths over the last 4 to 5 decades. So what do we mean when we're talking about asthma? Asthma is actually derived from a Greek word meaning to pant or to breathe very shallowly. And this is a typical breathing pattern for an asthmatic, and they often make a noise which we call wheezing. And that's often used as a clinical diagnostic tool for asthma. But it's a chronic inflammatory disease of the lung. It manifests as these recurrent episodes. And we know some of the triggers of these episodes, but some of them we're not really sure what triggers them. But this happens over and over in individuals. It's not something that seems to be consistent. They'll be fine for periods of time and then have additional episodes. And when they have one, then what they feel is their chest gets tight, they can't breathe, or they cough. The pathophysiological manifestations of this are airway hyperresponsiveness. This is the way that you actually diagnose an asthmatic. Because the reason they're not able to breathe is their airways are constricting. And let's see, I was told to use the pointer on the podium, but that's always a little bit tricky. I think I'm going to break the rule here. And they told me they were recording it, so they wanted me to use that. It usually advances the slides, so I think I'll try this. So what you see is this is a normal airway or upper airway, and this is an asthmatic airway. And you can see that the lumen of the asthmatic airway is quite narrowed. And this is due to smooth muscle contraction as well as mucus production and fluid accumulation in the airways. And so there's less air that can actually travel through these airways. To assess this clinically, and something we also do in our murine models of asthma, you give a natural bronchoconstrictor to cause the airways to constrict. But in normal individuals, you need maybe 1,000-time higher dose to get an actual constriction than in an asthmatic. So really you're looking at the sensitivity to a bronchoconstrictor like acetylcholine. And this is called hyperresponsiveness. It's the main pathological feature of asthma. It's also associated with an inflammatory response, and this is predominantly lymphocytic and eosinophilic inflammation, although I'll preface that in a few slides to tell you that there may be some diversity in that response. You can have elevated IgE levels, and this is called atopy, or the tendency to develop this type of antibody response is called atopy. There's a remodeling of the airways. If you look on the left side here, this is a normal airway with a nice intact structure, and this is an airway from an asthmatic, and this is from a bronchial biopsy. You can see you lose a lot of the architecture, you've got a lot of cellular debris, and the lumen is actually full of cellular debris and mucus. There's a subepithelial fibrosis that occurs right around the airway, and the muscle thickens, like is shown here in the illustration, so that the muscle can contract to a greater degree, but it is also actually thicker. And so when it gets a contractile stimulus, the lumen is narrowed to a greater extent. So this came out in Newsweek. It was the cover of Newsweek some years ago, but it highlighted that this was not just a clinical manifestation, that actually this is a recognition by the general public that there's been this increase in asthma. And they're saying here the scary spread of asthma, how to protect your children. So between 1960 and 2002, the increased number of cases, 72%. That's a dramatic increase. But when I first came to Hopkins, most of the pulmonary physicians that I worked with at that point in time had been here for a while, and they said when they first started their careers, they didn't see asthma patients. but it's become the dominant diagnosis or the dominant patient in the pulmonary groups. And so there's definitely been in our lifetime this huge increase in disease. So which groups of people are actually affected by this? On the left-hand side, this is age group and sex, and it turns out that the highest rates, particularly over 2001 to 2009, are in children rather than adults, and in females. And in terms of race or ethnicity, this tends to be mostly driven by inner-city African-American children. So this appears to be associated with changes occurring in inner city, or if you will, urbanization. So this is not only confined to the US. This is a map showing prevalence rates, and the red is greater than 10% or greater. And this, of course, is North America. It's quite red here, you see. And this is parts of South America, the UK, and Australia. It's interesting the bearing that has on it. But the one thing that it does show you is that in the more developed countries, you have much higher rates. You can see in some parts of Africa and others, there's basically no measurable asthma. So this is a disease of developed, industrialized countries. Now, asthma, this trend in asthma rates is not alone. You can see here over the same time periods from 1950 to 2000 that you've got other inflammatory or immunologically mediated diseases rising in the same time period. MS, Crohn's disease, type 1 diabetes. What I'd like to convince you by the end of this talk is that there may be some common elements to why this is occurring, even though our immunologists in the crowd will generally say, well, these have very different immunological profiles. But I'd like to convince you there may be some common elements to these increases in disease. The other thing you'll note is that many of the infectious diseases have been dropping over that same time period. Now, most of that is from vaccines and enhanced medical care. And some people who are anti-immunization like to draw this parallel, but I'm not a believer in that. I think we don't want to turn back the time and go back to a time when these were really major problems. So I'm going to try to explain perhaps why asthma, and then by inference to these other diseases may have some common elements in our lifestyle. So we don't really know what the etiology of asthma is, but we do know there are genetic components. So if you have a first-degree relative that has asthma, your probability of having asthma goes up considerably. One thing that's quite interesting about that is that it's a higher probability if it's your mother who has asthma rather than your father. So there's this maternal influence. And this was noted many years ago before we knew anything about epigenetics or other ways that maternal influence might impact on disease. But this has been repeated numerous times in many studies, and it's a pretty clear pattern. Atopy, that tendency to make the IgE antibody, is also genetically controlled and a risk factor for asthma. In terms of specific genes, there are about 22 different chromosomal regions that are associated with disease. So it's very hard to say it's this gene or that gene. It's clearly complex, and there are many different genes that are involved. But there clearly is a genetic basis. The fact that in identical twins that there's no concordance between the development of asthma in twins tells you there has to be environmental factors, and indeed there are. We think because of the type of inflammation that you see, where there are Tcells and eosinophils, that this is likely driven by some maladaptive immune response. And unlike autoimmunity, where you're responding to some self-antigen, this is generally driven by something in your environment, outside your body, something that you're exposed to. Some of the triggers we know for asthma are allergens themselves. These are called allergens because they induce this type of allergic response. I'm not going to show you today, but we've done a lot of work on trying to determine what makes a particular protein allergenic. Why in the millions of proteins out there, why are there a select group of proteins that tend to be allergenic? And we have some very exciting data on that, but I'm not going to focus on that today. So viruses usually will cause exacerbation of asthma, particularly this time of year. Most people that are asthmatic, when they start getting colds, get into the cold season, they will have a greater number of asthma exacerbations. So we know that certain viruses can make this worse. There are certain viruses like RSV that there's some debate about whether they just exacerbate or whether they're an actual inducer of disease. It's sort of a chicken and the egg question that really hasn't been resolved. But what is known is that 100% of you are positive for RSV by the time you're a year old. Okay, but there's a handful of kids that actually get put in the hospital with severe responses, and those children go on to have asthma. So there is this very tight association between that early life exposure to that virus, that particular virus. Pollutants, and I'm going to talk about air pollutants in a few minutes. Maternal smoking is also a risk factor for development of asthma. Diet and gut flora, which I'm going to spend the majority of the talk talking to you about some of our new data on the role of the gut flora. Sedentary lifestyle, and Mike was talking about some of the talks yesterday on micronutrients that I heard, and it turns out that vitamin D, many of these things actually regulate the immune response, and that may be how they're actually working. And so definitely in children today, they don't get exposed to a lot of sunlight. They're never outside. I know myself, I'm much paler than I was 20 years ago because I never get out of the building. But anyway, so in the late 80s, there was a landmark paper that really showed that different CD4 T cells could be subdivided and that they had different functions in the immune response. So Th1 was characterized by interferon gamma production, and this was thought to control organisms that were intracellular pathogens that would infect cells and be inside the cell. Th2 was the opposing pathway that was first discovered, and this was thought to deal with extracellular organisms like large parasites and worm infections. Okay. One thing to note about the Th2 response that's relevant to asthma is that this response is necessary to protect the fetus. And so there are high Th2 cytokine levels in the placenta during pregnancy because it opposes this Th1 and your body recognizes the fetus as a foreign object. And so without this Th2 response, it causes spontaneous abortions. And so this is critical for maintaining the fetus. It's also true that that means that when the children are born, they are bathed in this Th2 environment. And at some point, that needs to be balanced and corrected. And that's what we think that the environmental exposure to different microbes in your environment do. But this is characterized by IL-4, IL-5, and IL-13. Parasites work through this particular pathway. They induce this pathway, and this is what helps clear parasitic diseases. The interesting thing is that a parasite, by definition, doesn't want to kill you. They actually want to live in harmony with you. And so even though they evoke this type of response, they also evoke this response called T regulatory cell. And Tregs basically are suppressive, and they will suppress both a Th1 and a Th2. So normally what they do is try to balance the immune response. Once you've mounted this big response and you need to then dampen it, the Tregs will do that. Okay, this is activated by vitamin D. It's actually, this was, I think when people were trying to determine how they could grow these in vitro, they just went to the Sigma catalog and pulled out everything, and they discovered that vitamin D was a very nice activator or stimulator of Tregs. This population is characterized by IL-10 and TGF-beta. It is associated with tolerance to exogenous allergens. And as I said, it's induced by parasites and commensal bacteria so that you can live in harmony with them. Because when you have a Th2 and a Treg response together, you allow that parasite to be in residence without mounting a really aggressive response that harms both you or the parasite. Oops. Okay. So as I said, this population inhibits both of these others. So how do these Th2 cytokines actually induce asthma? We worked on this for about a decade. Our group was one of the first to show in animal models that the CD4 T cells were absolutely essential. We used antibodies at that time, but this has been replicated in many, many studies since then. We worked on how these individual Th2 cytokines could play a role, so IL-13, IL-4, and IL-5. IL-13 and IL-4 induce the atopic type of antibody, IgE, and it activates mast cells, and most of you probably have heard of histamine, leukotrienes. The IgE binding the mast cell will release histamine and other bronchoactive substances from the mast cell, and then it constricts the airways and causes fluid accumulation. Eosinophils are strictly driven by IL-5, and they release specific mediators as well, but even though they appear to be always present in many asthmatics, we don't really know what they do yet. After many years of studying that, there's a controversy. There was a clinical trial blocking IL-5 and it failed. It didn't work in asthma. So the jury's out on what that population does. So one of the things that we had shown is that IL-13, this particular Th2 cytokine, was the central mediator, and it by itself could induce the entire response. You didn't need any of the other factors. And I'm showing you that here. This is our measurement of airway hyperresponsiveness that I told you about. We actually cannulate the mice and ventilate them and induce bronchoconstriction with acetylcholine. And when you just give the acetylcholine, this is the response of a normal mouse. This is airway pressure changes in that catheter. If you give IL-13 to a totally normal mouse, otherwise with no antigen, they become hyperresponsive. So you don't need any of the other cytokines or any other antigens. This cytokine alone can do it. And you can see it's a very potent inducer of mucus production because the mucus is stained in the pink color here. You could see just giving that cytokine, you get tremendous goblet cell hyperplasia and mucus accumulation. And this is staining, fluorescent staining for eosinophils. They also accumulate in response to this particular cytokine. So this particular study, which was published a couple years before I left here, has fueled many, many studies in asthma and clinical studies as well. So is that pattern actually important in humans? And I put some human data in here for those more interested in that. And there was an original study done back in early 1995 when some of those other cytokines really weren't even available to study because there weren't reagents. So the first study was done in a Tucson cohort study, longitudinal. It turns out that many years ago, people who were allergic or asthmatic, they used to tell them to move out to Arizona. So they have this huge population that's just been interbreeding, I guess you'd say, and lots of asthmatics. You know, unfortunately, they brought all their allergens with them. They've tried to plant grass and trees and things in the middle of the desert, right? So they had a window of time when they were pretty good, but now they're back to suffering from asthma because they brought all of their triggers with them. But anyway, there's a group there who's done a fabulous job of following many of these kids over long periods of time. And what they found was that Th1 cytokine production, they measured it at 9 months of age in these kids. And typically with asthma, you can't really diagnose it definitively until they're about 5 or 6 years of age. So this makes these studies difficult. But what they did was collect these PBMCs from their whole cohort, and then they waited until these kids were 6 years of age. And then what they found was that those that didn't make much gamma, or that Th1 subset, actually did have a higher rate of atopy or allergic responses. And this also correlated with their parents. And I've already told you that this is a genetic condition, and if you have a parent who's atopic, you're more likely to be. But the take-home point I wanted to make here is that they could tell at 9 months of age that their immune response was already abnormal. Okay, so this is a very early event. Things are happening early in the life of these kids. They didn't have to wait for them to be 6 years of age to diagnose them because the changes occurred very early. The next studies showed the same thing. They took cord blood cells, actually, of offspring from atopic mothers. So they recruited atopic mothers during pregnancy, and they took the cord blood when the child was being born, and they found that suppressive subset Tregs were down. They weren't there in numbers, and they weren't really functional. So they've lost that regulatory arm. They've lost the protective Th1 and the regulatory arm. And they saw that they had high IL-13 levels. Okay? So together, these suggested that these CD4 T cell cytokine patterns were established very early in the lives of these asthmatic kids or atopic kids. And that what seems to be occurring is there's this imbalance between the Th2 and the regulatory arm. Okay? So I told you there was a lot of studies fueled by that original IL-13 paper, and I'm happy to say after how many years now? This is published in 2011. The original study was 1998. They finally have done a clinical trial that was done in a reasonable fashion that actually shows a benefit of blocking the IL-13 pathway. For those of you who have worked with drug companies, you know it can be quite maddening to get through their process and for them to move forward on things. But what they did here that was unique and different from some of the other studies that had been done is they actually looked, which seems intuitively obvious, but they looked to select people into their study that actually had this pathway upregulated. Okay? You would think that if you were going to block a particular mediator, you'd want to know that it was actually present in the population you were studying, but it hadn't been done in previous studies. So they selected a group that had Th2 cytokines and a gene here called prostin that was a marker of that IL-13 pathway. And so when they selected those and they treated them with this anti-IL-13 antibody, they saw an improvement in their lung function. Interestingly, though, the ones that did not have this fingerprint, if you will, had absolutely no benefit. So this suggested two things. If you treat the right subset of patients with this antibody, it's going to be effective clinically. And so more studies are being done to replicate this. But it also suggests that there's this group here that isn't a Th2 skewed immune response, and that this treatment wouldn't be effective for them. So this type of approach, I think, is one that's gathering steam in many clinical trials to really hone in the right population and show that the thing you're trying to modulate is actually present. So with that, I think we can agree that the disease must be heterogeneous, because you've got a group there that doesn't respond to that blockade at all. And in fact, it is. We know the age of onset, childhood onset of disease versus adult, those are probably totally different diseases, but have the commonality of having the impaired lung function. There are some asthmatics that are non-allergic, so they don't have elevations in IgE, and that's probably a very different type of disease. There are differences in the type of inflammatory response. As I told you in the beginning, we usually think of it as an eosinophilic disease. But it turns out that there are more severe asthmatics that have mainly a neutrophil response, not eosinophils, or they can have both. There's differences in severity. The interesting thing about this disease versus autoimmunity, where most autoimmune patients tend to escalate and their disease gets worse over time. This doesn't turn out to be true with asthma. If you're a mild asthmatic, you generally stay a mild asthmatic. If you're severe, you're severe. So that suggests that these are different pathways to development of disease, or there are different things that may be driving them, okay? And there are differences in drug responses. So the mainstay therapy for asthma these days is inhaled steroids. Okay, now obviously those aren't optimal because they have many side effects. But the thing about them is they're cheap, easy to come by, and so they're commonly used. But it'd be nice, particularly in children, to have other drug possibilities that you wouldn't have the side effects on growth and bone development from inhaled steroids. But there are many of the severe asthmatics that even at the highest possible doses, they don't respond, they don't get any better. So it suggests that maybe they have a different disease process, or that they have some abnormality in the glucocorticoid receptor pathways. We think it's a different disease, and I'll give you a little bit of data to maybe support that. So I told you about these two opposing pathways, Th2 and Tregs. More recently, there's another T cell subset discovered called Th17. And this is associated with TGF-beta, IL-6, IL-1, and IL-23. It's the family of cytokines that regulates neutrophil maturation and recruitment into tissues. And it's been highly associated with autoimmune diseases. So early days, we used to think all autoimmunity was Th1 mediated, but now we know that it's probably more likely to be Th17. Okay, so there are some recent studies in asthma that suggest this more severe phenotype, or the severe asthma, is probably driven by the combination of these two. And the data for that is shown here. A group in Canada very nicely showed in bronchial biopsies of asthmatics controls, mild, moderate, and severe. So you can see the number of cells that are positive for these Th17 cytokines is greatly increased in severe disease versus those with no disease or mild disease. And that was true for the other family member, IL-17F, as well. So in human studies, this is the best you can do. You can't really prove that these pathways are involved until you have enough data to actually do a clinical trial. So in our lab, we actually test these hypotheses in animal models and provide data for clinical trial down the road, hopefully. So what we had found, and this was relevant to that article that was published on the different mouse strains. So years ago, when I was first here at Hopkins, I had done this complete survey of different inbred mouse strains and shown that there were strains that were highly susceptible and then those that were not, okay? And at that time, they were associated with differences in these Th2 cytokines. The resistant strain didn't make Th2, and the susceptible did. But taking those same mice from the same supplier and just moving them over to Ohio, they totally changed in their response. But what's even more curious, coming back, they're back to their old response. So, 14 years later, the same phenotype manifests again when being in a different animal facility. But in the meantime, between those moves, we actually found that our most susceptible strain, the AJ, in terms of airway response, made the IL-17 family members, and that susceptibility was associated with a high Th2 but a Th17 response as well, suggesting that it's the severe phenotype. You need both of those arms of the immune response. And so we tested that by blocking that with an antibody to IL-17A, one of the family members, and we saw we could significantly reduce their hyperresponsiveness. It's not entirely back to normal because of this elevated Th2. Okay? So how could this be? What's driving this? And I'm going to show you this because I'm going to use it again in a minute in context of the microbiome. But we know that T cells are, as my husband would like to say, dumb as dirt. They don't actually direct anything. They are told what to do by these dendritic cells. All their signals for what to respond to, how to respond, come from a dendritic cell. And Ralph Steinman won the Nobel Prize last year or the year before for discovery of this particular cell type. And it is the main player in terms of driving the type of T cell that you might develop. So Th17 cells require IL-6, TGF-beta, IL-1, and IL-23. So we looked in our 2 strains of mice to see, are there any differences in these cytokines? How could they be developing a Th17 response to the same stimulus when the other strain does not? And as it turns out, there's a big difference in this one mediator here called IL-23 between the 2 strains. The others really aren't that different. And so we think that this is the major driver of this more severe phenotype in the Th17 arm of immunity. So returning to the initial thoughts about why is asthma increasing? Okay, I've told you all these different triggers, and many of these are certainly associated with the modern westernized lifestyle. Air pollution, cigarette smoking, diet, sedentary lifestyle, playing on your computer the whole time, indoors, and not getting outside. So here, before I had left here, Pat Bricey and I had worked on air pollution, and it turns out, at least at that point in time, Baltimore had quite a high air pollution as well. Not as bad as these. This is a recent photo taken in China on a normal day. Now, if any of you have visited there recently, you don't have to be asthmatic to get chest tightness when you walk down the street in this kind of air pollution. It's really striking, and your chest does tighten up, even if you're not prone to that type of thing. So Pat, through the Children's Health in the Urban Environment, we had a project where we worked on air pollution here in Baltimore. One arm of it was looking at the children in various neighborhoods around Baltimore, and the other was to look at what did these pollutants actually do? What could we determine the mechanism of how they might induce asthma? So now this is a Mac to PC conversion here. Strange numbers. Anyway, there's lots of data done in the U.S. showing that there's an association between exposure to that type of air pollution and asthma, both for increased hospitalization, asthma medication use, and decrements in their airway function and changes in their airway structure as well. There's a recent study that was done, and actually one of our recruited faculty from Cincinnati actually worked on one of these projects. And they showed that exposure to traffic-related particles during childhood is associated with wheezing when you get a little bit older. So they did a mapping study where they looked at air pollution in Cincinnati and mapped the amount of wheezing in children over time and how close they lived to the actual busiest airways, roadways. And they found there was a nice correlation between that. And Winnie Tang in our group actually did this study with them. She went on to show that polyaromatic hydrocarbons that are present in that air pollution were associated with the hypermethylation of the interferon gene. And I told you that interferon was something that would protect against the allergic response. And this was done in cord blood of children in that cohort. And this suggests that there's epigenetic fetal programming early in life that's actually predisposing these kids to development of asthma by being exposed to the traffic and living close to these heavy trafficked roads. So what happens in animals? So Pat had set up this system. It was a pump outside of the window of the school here. And we collected air just right off the street. And it was put through a cyclone to size it in the size particles that normally are associated with this type of response. And then we took it, concentrated it, and gave it to the mice. And you can see here that they became hyper-responsive after that exposure. This was, in these cases, just a single exposure. Okay? And you can see the type of inflammation that developed in their lungs. It was pretty dramatic. And actually, these black dots are part of the particles that actually came through the air and deposited in the lungs of these mice. And there were these Th2 cytokines were elevated in the lungs of these mice. And we went on to show in a much later study that this was all T-cell driven. Now, we don't really know what component of the air is actually activating the T-cells. We're a little confused by that because diesel and some of these things aren't really your typical antigen, but something in this real-life gamish is actually activating T-cells, and that's why you're getting the Th2 response and development of asthma. So what other triggers or lifestyle things are associated with the increase in asthma in developing countries versus developed versus developing? And there was a hypothesis put out in the 1960s, late 60s, by a German investigator, and he called it the Hygiene hypothesis. He actually based it on the fact that he had noticed in his practice that there was more hay fever or allergic responses in families that had very few kids. So one of the changes you see in the modern world as family size is reduced. His thought process was that the fewer kids you had, the fewer infections you had, because you weren't passing it. You know, if you've got 8 kids and they all come home and swap germs, as they tend to do, that you would have more infections. And he thought the infections, the larger family sizes, were actually protective. And what he noted was it was only the families with one child or small families that actually had more allergic disease. So that was put forth back in the 60s, and it's been tested over many years. And some parts of it actually make some sense, but I'll tell you how we're interpreting it now in 2012. But one of the ways that people have studied this is comparing the conditions in the modern country, developed countries versus developing countries. And things you see for sure are larger family size in non-developed countries. They live on farms. There's a difference in their intestinal flora. There's low antibiotic use relative to here, and poor sanitation. All of those would lead to increased microbial exposure. Okay, I'm not saying what type of microbes, but just in general, more microbial exposure compared to westernized countries with smaller families, a very different intestinal microbiota, a high antibiotic use. Just working at a children's hospital before I came here, pediatricians who were seeing kids, now of course they were in the hospital, they were seeing kids that had had 4 or 5 courses of antibiotics before they were 2 or 3 years old. That just completely destroys your gut microbiome, you know, very early in life at this window when I'm trying to convince you is very important. So there were differences here, and many studies had been done to look at, to find a specific infection that might be driving this. And there were early studies done with tuberculosis, with other things, and nothing so far has really panned out. But if you look closer to home, and you look, as I told you early in the talk, that we have higher rates of asthma in inner city versus in, outlying communities. And some very elegant studies have been done in Germany where they show if you live on a farm, and these studies were done of Germans living in cities versus those living in farms, so they're genetically, somewhat close. They find that those living on the farms, and their definition of farm is where the cow is pretty, is attached to your house or in your barn. And those families, they tend to be protected from development of asthma. If you actually have a dog in the house early in life, you're exposed to quite a bit of fecal material, even though you don't like to think that you are. But you're actually protected. This doesn't apply to cats. Cats are very allergenic no matter when you have them. The attendance at a daycare is actually protective. So for those mothers who are worried about sending their kids to daycare, this is actually a good thing. And what they found in all these studies is that these exposures that occurred in the first year of life had much stronger effects, protective effects, than if they occurred, when they're 10 or 12 years old. So what would be the mechanism of this type of thing? How could we envision this hypothesis being true? And it turns out that the more microbes you get, you would induce the gamma response or the Treg, and then it would inhibit the Th2 arm. So that's been the theory for how this might work. I'm going to skip over this in the interest of time. So one thing that's come up recently, and I think every seminar I've been to in this room over the last month or two has mentioned the microbiome. So this is definitely an emerging theory for many different things and of interest. And one thing that came out of the studies comparing countries overseas from the US and other North American is that there are very different patterns of bacterial colonization in the guts of people living in westernized societies versus those living in Africa, both quantitative as well as what type of bacteria. And in the Western countries, we tend to be colonized with gram-negative bacteria much later in life. So there tends to be a preponderance of gram-positive early in life. And the other thing is that in the developed countries, the same strains of bacteria tend to take hold and they don't vary much. Once you've kind of established your flora, it stays. But in developing countries, there seems to be a lot more diversity and a lot of changeover in the microflora. So there are a number of lifestyle issues that have been associated with asthma. So surprisingly, even though this is a very short exposure, the increase in cesarean sections is associated with increased asthma rates. And this is because that very early colonization with bacteria seems to be critical. And this has been shown in numerous studies now. Doesn't appear to be a fluke. It's very reproducible. And as everyone appreciates, I mean, now you can just say, well, I want to have my baby on the 21st of December at 8 o'clock. So there are many cesarean deliveries in the Western societies now. Increased antibiotic use is very highly associated with increased asthma rates, and you see that there are particular bacteria that are outgrowing from this. C. difficile. Decrease in breastfeeding also seems to have similar effects and is associated with risk factor for development of asthma. So the microbiome, what that really means is you're looking at the communities, not at a single organism, but the communities within the GI tract. And this has been made possible with better technologies where we can actually assess, not by taking samples and culturing them, but looking at the sequences of large numbers of different bacterial strains. And then we can do this simultaneously. And what was done here, this is looking at the GI flora between a non-allergic person and an allergic group of children. They're 5 years of age. And you can see there's some pretty big changes in a couple of these populations. So in the allergic person, you have more Clostridia and less bifidobacteria. And the reverse is true in the non-allergic. So there are shifts in certain populations. And to date, we haven't really gotten down further than this to see exactly what those organisms are within those broader categories. But these shifts seem to be associated with disease. Now, recently, they've also looked at the lung microflora. And I was always taught that the lung was pretty sterile, but it turns out that it's not. It says if you look at this, you've got all the same families of bacteria that you had in the GI tract. So that was quite curious. But this, because these are all done by sequence, you don't know for sure if they're colonization or whether you're just inhaling debris or parts of organisms. But it's still pretty striking that you have all these organisms in the lung. And again, you see these same shifts in decrease in the Bacteroides and increase in these gram-positive bacteria associated with asthma. And this is in bronchial biopsies of both adults and children. So all these studies together have led to the hypothesis that the change in gut flora or the balance in the communities within the GI tract may be associated with disease development. About the time we were getting interested in this, a group in New York had the similar experience to us about their different mice having very different immune responses. And so they published a very nice study showing that they took this black 6 strain of mice, same ages. The only difference is if they bought them from Jackson Labs versus Taconic, they mounted a Th17 response or they didn't. Here they didn't, there they didn't. They did a very nice microbiome type study and found that it was due to this single organism that was different between the mice grown in one facility versus the other. And genetically, they were identical. So this had to do with what was in the facility. And it turns out now they've figured out that it's probably the diet that they're feeding these mice and the different suppliers. But what they found was there's one organism called segmented filamentous bacteria, and you can see it. This is a micrograph of the gut epithelium. There are these filaments here that are just completely covering the GI tract, and they're quite invasive. If you see the cross section of these, it looks like seaweed floating in the ocean, just completely covered. So this organism happens to belong to that Clostridia family, and it's quite unique. It has never been cultured, and no one has ever been able to culture it. So in clinical studies, when you're looking at, bacterial infections, you could not detect this because you can't culture it. It turns out it's like a parasite, and it cannot grow on its own. Recently, the sequence has been found out, and it turns out that this particular organism is missing a number of genes that will allow it to produce energy on its own. So it's totally dependent on being in the gut epithelium for nutrients for survival. And so you can see here it inserts itself in the gut epithelium and survives here in this niche. But bacteria compete for those nutrients within the GI tract. And once one is established there, it takes a lot sometimes to get rid of that one and replace it with another. This isn't known to be pathogenic in the classic sense. One thing about it is it's completely cleared from animals, both mice and humans, when IgA antibody develops. So very early in life, this will be cleared. IgA knockout mice cannot clear this, but if IgA is present, this organism will be eliminated very early in life. It's been shown in a couple other studies to be associated with autoimmune disorders, EAE, inflammatory bowel disease, and arthritis. So here we're showing one organism that could induce many different immune disorders that on the surface don't have anything in common. So we took advantage of that, and we asked if in fact that same infection could induce asthma. And so we exposed them to our allergens. We got the mice from the 2 different suppliers, and this was our gram stain to prove that they were there. And by sequencing, we could show that only the mice from the one supplier, Taconic, had this organism present. Okay, sorry if we're running over here. When we looked at their airway response to acetylcholine, we can see that the ones that have this organism present are twice as responsive than the other. And remember, these are genetically identical. We've treated them in exactly the same way. The only thing different is where they lived in early life. Okay? They have a neutrophilic response, which I told you earlier was associated with more severe disease. And there was no difference in their Th2 arm of their response or their IgE. But what they did have was this Th17 arm. So we think that this organism, this imbalance in the GI tract, is driving this more severe phenotype associated with the IL-17 pathway. So I told you a minute ago, this organism is cleared. In the mice, it's cleared by 12 weeks, but you see that the Th17 response persists out as far as we've measured it. So what that means is that, you can have this early in life and clear it, and you're still going to have the consequences of that with this type of immune response or deviation persisting. So that study doesn't really prove that that's the organism. So what we did, it turns out you can actually get this organism, SFB. There's a group in Japan that have made a mouse that's only colonized with that organism. And they will gladly send you the fecal pellets, okay, that contain only that organism. So we got these fecal pellets, we actually gavaged the mice with them. And we take the mouse that's from the supplier that didn't have this present, and we give only this organism, okay? What we see is that, in fact, you can transfer it. So you can take a mouse who wasn't grown or early life in that, you can give them this organism, and in response to that, you become hyper-responsive with this Th17 arm of the immune response. So showing that it's this single organism that can induce this response. Okay, we blocked IL-17 in these and showed that, in fact, this increase in the hyper-responsiveness was due to the IL-17 pathway. So it connects the SFB and the Th17 arm. So I showed you this earlier. What we wanted to find out is it similar to what we had seen before, that this pathway might be where that organism is working? And it turns out that, in fact, it is. You have, in the mice that have SFB, you have more IL-23 driving the Th17. But here you have a lack of that Treg modulation. So what this does is shift that balance to the more pathogenic arm. So again, this phenotype, this imbalance in the Th17 promoting and Treg promoting persists out to a later age after this organism is cleared. So this suggested that there might be some changes in the bone marrow where these cells actually arise. And we looked at this, and we took bone marrow cells from either the mice that were coming from the supplier that didn't have any contamination with this organism, or the one that did. And we transferred those to a mouse that had no SFB, and we asked, could those single cells actually transfer that susceptibility? And it turns out they can. If you took the bone marrow cells from the mice grown in the colony with the SFB, they're much more hyperresponsive. So this says the action is at the bone marrow dendritic cell level. And here again, it's with Th17 cytokines. So what we thought was this organism only lives in the gut. Now, I forgot to mention earlier, it's anaerobic, so it does not live in the oxygenated lung. It's only living in the GI tract, and that's the only place it's been found so far. So it suggests that organism in the gut is producing some signal which is changing something in the bone marrow. Okay, so we look to see what that signal might be. And we did a RNA-seq heat map of the GI tract of these two different mice. And there was one huge hit here. It's called serum amyloid A. And for the clinicians in the group, they've probably heard of this before because it's highly associated with a number of inflammatory diseases such as obesity, cardiovascular, and it's known to be a risk for many inflammatory conditions. It drives Th17 and a severe asthma phenotype, and it induces that mediator IL-23. What we saw was the mice that we had transferred that SFB to had more of this in their serum. So we could show that it was being released from the GI tract into the serum of these mice. But this is a tricky mediator. It turns out it's quite promiscuous, and it seems to activate a number of receptors on immune cells. The classic toll receptors, the CD36, and this pathway called FPRL1. So what we did was to take a look to see if there were any changes in these genes epigenetically in the bone marrow. to see if, in fact, we could identify which of these receptors might be the key. Because I told you that early infection in the GI tract was changing the bone marrow, and it was persisting over time. So it suggested it has to be epigenetic. So we did that, and this was a targeted epigenetic look. We didn't do the whole genome. We have done it since, but we started with these targeted genes to see if their chromatin could be remodeled such that there was activation of these genes. And in fact, we found that the FPRL1 gene was in fact in an active configuration in the mice that had the SFB infection, whereas the other receptor for it was actually turned off. So this gave us a hint that perhaps this might be the key pathway. What is this thing? Most people haven't heard of it. It's a formal peptide receptor like 1. It's a chemoattractant for many inflammatory cells, including the ones that we care about, the monocytes and the T cells. And it's chemoattractive. It mediates the activity of the serum amyloid A, so that puts the two together for us. So we gave the dendritic cells from these mice the serum amyloid, and we could see that only the ones from the SFB infected actually responded to the stimulation, suggesting that in fact the receptor was more highly active compared to the other strain. Blocking it with the specific inhibitor only affected the mice that were infected with the segmented filamentous bacteria. So I'm going to skip through a number of things here and just cut through the chase to tell you that the fact that we could take bone marrow cells from a naive mouse, from the mice grown in a facility with SFB or not, and transfer asthma suggested something had to be happening in the populations in the bone marrow. And in fact, we found that that was true. The population that gives rise to macrophages, monocytes, dendritic cells was upregulated in those mice that had this gut flora. Okay. And the population that branches from there, so this is the one I just showed you that's upregulated. The branch that gives you platelets and red cells is actually reduced because they come from a common progenitor and there's a balance between them in terms of hematopoiesis. So having this single organism in your GI tract can change the composition of your bone marrow, which is a pretty striking finding because these changes can persist for quite some time and really control the response to anything that you're exposed to during that period of time. Okay. So this is our conclusion, basically, that we have found that this single organism that is colonizing the GI tract can drive this Th17 response in the gut, but it releases something into the serum that changes the set point of your bone marrow so that it now is conditioned to drive that subset of T cells, not your protective subset, the Tregs or others. Okay. And this was quite striking to us because it suggests that this is a long-term epigenetic conditioning, and we showed that that was associated with epigenetic changes in the bone marrow cells. So we think that this is going on when you had gotten exposed to this organism in the GI tract. Then when you get an exposure in another organ like the lung or the skin, that same subset is now coming into that organ because that's what your bone marrow is promoting or producing. So this could also suggest that this could drive all of those autoimmune diseases that I told you about. It can drive asthma and the autoimmune diseases as well because they're all Th17 mediated. So is this important in humans? That's what we always want to know. We don't know the answer to that, but when we first started this project, it had been said that we never see this organism in humans. Well, it's because you can't culture it. Okay. In most clinical studies, you only know what's there by culturing. Okay. This thing cannot be cultured because it's a parasite and can't live on a plastic dish. But when they've done sequencing recently in 2012, a large number of people, and they weren't looking for disease, they're just looking for this organism. They actually find that it's present, but it's only present in kids younger than 3 years of age. So you clear it when you have normal IgA. When your IgA develops, you clear it, but it has long-term consequences. And so many groups now are starting to look to see in populations with different diseases whether this tracks with these particular autoimmune and allergic diseases. Our data would suggest that this organism makes epigenetic changes in the bone marrow, which could persist for quite some time. And then when there are threats in different organs, the Th17 arm is activated and results in various diseases. We suggest that this might be relevant to more than just asthma, because the Th17 arm is part of MS, diabetes, arthritis, Crohn's, and obesity. So whether it's this particular organism or others similar to it, or a combination of organisms, we think this data is pretty strong in suggesting that these diseases, which I showed you, they were all showing the same trajectory in terms of increases, are unified by these changes in the microbiome. And so we look forward to seeing other studies in these particular diseases to see if in fact this is true. And I know there are many people looking at microbiome analysis in many different diseases. I think we're going to find that certain categories of bacteria are clearly associated with these increases. And these things are very susceptible to many of the dietary changes and things that you see in the Western world. So this is my last slide. I showed you that rural living, exposure to dogs and fecal material early in life probably induces this very balanced gut flora, induces these T regulatory cells that can suppress inflammation once you've mounted appropriate responses. But in our Western way of living with maternal atopy, cesarean births, antibiotics, high-fat diets I didn't talk about, but this also has a profound effect on the gut microbiome. With a genetic predisposition to a Th2 response, you get this intestinal imbalance. And then when there's a challenge to the lung or another organ, you mount this Th17 response, getting, at least in our model, a severe phenotype. So again, I think this has broad applicability to many different disorders that are associated with these immune responses. And I think the good news about this is epigenetic changes can be reversed. And we can change many of these things. We can change the way kids are fed and what they're exposed to early in life, and that perhaps we can reverse some of those trends and increases in all of these diseases. So I will stop there and thank my lab group here at Children's and then here at Hopkins, and Pat Bricey also for the help with all the air pollution studies. Thank you so much.
Thanks, Marsha. So I'm sensitive to time, and I know some people may have to leave. Maybe I'll just ask one question, and then we'll break for the reception. But what's the mechanism by which SFB infection causes the epigenetic changes?
Well, we think it's through the serum amyloid and serum amyloid altering its receptor, the FPRL1. We don't have the exact mechanism, but we're putting the pieces together at the moment.
Great. So unless it's burning, let's move in and talk to you out over a glass of wine and some good healthy antibiotic or